Chances are that you won't get the certificate. So please try to, while I am going through this, please try to revise and ask yourself if you're clear with that. So
first, if I am to ask you, why omics? This is going back to the first lecture that Professor Sanjeeva gave. What's the point of studying omics? We are very happy, right? We were studying genes in isolation, proteins in isolation, and we have been doing this for a long time. Why do we need to study? Why at all go through all this headache? I mean, it's much easier to do a small test, maybe check for interleukin, or maybe check for anything. Why do we need omics?
So because eventually the whole pathway is regulated by all proteins and genes and mRNA and microRNA, etc.
Right.
It's like a whole complex. If studying a singular entity will not define the whole biological aspect of how the modulatory role is going on.
Right. So that's what happened in the 1990s, and this is how this lecture would go. So initially there were departments of genetics, department of biochemistry. So those who are from biochemistry, forgive me, but this is what started off. In all universities across the world, zoology, botany, these are even older, ancient departments in science. Then there were genetics and biochemistry, and they started looking things at isolation. But if you look at it, any system does not work in isolation. A very simple example, suppose there is no railway in India. Would you be sitting here? Many of you will be sitting here, right? And you can tell that there are n number of reasons why, because you will be able to travel here, right? Most of the thermal power plants run on coal that is brought via railway network to these thermal power plants, and these electric fans and ACs, they won't work. So we are a network. We can say that we are all self-made and we are independent, but fact of the matter is, neither are we self-made nor are we independent. We are interdependent, and we are closely the way we behave, the way we move ahead, we are independent, we are linked to each other. And when you have to characterize multiple genes, when you have to study these impact, you teach. You have to learn from these multiple gene products, multiple genes. When you have to study these impact, you teach. You have to learn from these multiple gene products or genes. So when we are using multi-omics or omics approaches, our primary area is usually human health, disease, agriculture. And why we need to study these multi-omics is because DNA, you see, is very static. You're born with a set of DNA, and throughout your lifetime, might be there will be some epigenetic changes, but overall your genome does not change much. But you do, right? We are born very tender, young, and gradually we age, we have gray hairs and wrinkled skin, and one day we would die. So to study DNA genomics, it tells us that, okay, this can happen. If you look at the point of view of a disease, you can say if you have this, like going back to yesterday's lecture, so Angelina Jolie had a BRCA mutation, and she had a chance that she might have breast cancer. She went for mastectomy. She did not have. She might have had, but since it would be a triple negative breast cancer with very low poor prognosis, she went ahead to take a prior step. RNA, what it tells is what appears to be happening. At RNA or transcriptomics, they never tell us what is happening. It's more like what appears to be happening, because you see, even this year we have got a Nobel Prize for what? MicroRNA. And what does microRNA do? It regulates other RNA and even sometimes proteins. So it's more of a regulatory complex. So we know if you have a RNA or a transcript for a gene, it's very unlikely that whatever you see at the level of RNA, you would be seeing at the phenome level. You might see the same, you might see something extremely different. What primarily makes it happen is proteins and metabolites is more of a crime scene. When you look at metabolomics, you see it's more like a crime scene. The crime has already happened and you have metabolites all around. Then you start predicting that what has happened or sometimes what is in process, right? कहीं पे आग लगी है, धुआं चल रहा है, that is a metabolite, right? You don't know who set the fire, but you can tell definitely that it is in fire. So this is basically central dogma and how multiomics have evolved. And all of these work in conjunction with environmental factors. So you cannot forget. So it's part of our genome, the nature, and then you have the nurture. Together, they control. So proteomics was invented, the term was invented by Mark Wilkins in 1994 while he was still doing his PhD. So while those who are into PhD, don't feel disheartened. You can do good things. So Mark Wilkins currently is a professor working in Australia, and he discovered the term proteomics, and he was actually inspired by the word genomics. He thought genomics as a term had just come up, right? And people were talking of genomics in the early 90s. In 1994, proteomics burst into the fore. And I will show you over a period of time how things have changed. So he defined proteomics as the entire complement of proteins including modifications produced by an organism or a system. What he perhaps missed out is at that moment or time, because the beauty of proteome is it keeps on changing, right? When you wake up, you have a different proteome. When you go to sleep, your proteome is entirely different. So is your metabolome. Your genome and transcriptome, our genome does not change, then it would be a very difficult thing for us, right? The guys in forensics will be banging their heads. Crime हो गया, उसके बाद आदमी का genome change हो गया. You can't do that. So, but in proteome, it does change. It does change to even the smallest of stimuli. The proteome will change, and that gives us a very good way of understanding how events are unfolding in time and space. So I think I can go through these because you already know they serve as catalysts, they have signals, and we have gone through this. It's more of a repetition. So what I'm trying to tell you is how this field is evolving. So this is when the first term was invented in the 1992-93, right? And you see there are almost 0 papers. And then you see how they have just shot through the roof. This is 2023, and in 2024, perhaps once we end 2024, we can get the data for that as well. So proteomics as science has gone up, and this is like almost like I think 20,000 articles per year that mentions proteomics as a keyword. It's not proteomics articles that have used proteomics as such. Proteomics as a keyword means proteomics is a big part of that work that they have done. And now we have HUPO, Human Proteome Organization, that is working. Professor Sanjeeva is part of HUPO. We also have in India the Proteomics Society India. All of you many of you might know. Those who do not know, do Google them up, and they are a good group working and spreading the knowledge of proteomics. And when we talk of proteins, that molecules that do work, you can easily say that if this is the protein abundance and this is the mRNA abundance, they are intimately tied to the cell state. mRNA measurements are often it's not a true proxy, so they are more of a proxy when you measure mRNA levels or transcriptome level for any disease. You do not actually get the true picture, you get the proxy, right? You know, all of you are from college, you know what is a proxy. What is a proxy? Proxy लगा देना मतलब क्या होता है? False attendance. So proxy is not, so proxy लगा देना is of course false attendance, but proxy is like you are, see a strong correlation. So this is for the entire proteome, and what they did was you can look up on this paper, this in class biology, it's a very old paper, yet I don't know how stronger correlation, because techniques back then were not very strong. So there are two kinds of proteomics. Can you tell me what is discovery proteomics? You have gone through so many lectures. Where's the mic? Yeah, anyone? Yeah, please. Can you pass the mic?
Hello?
Yeah.
You are trying to study a protein that hasn't been discovered before.
No. It's a good try, but discovery proteomics. So proteomics studies can be of two types.
Data dependent and data independent.
No, that's acquisition. Yeah, Anurag.
Trying to find out the unknown target in discovery proteomics. So we don't know about the protein which we are looking for.
Almost very close. So since we don't have much time, let me break the news to you. So discovery proteomics, where suppose you are trying to understand a novel pathway, right? You have two groups of cells. One is a cancer cell that is responding to a drug treatment. Another cell line, it's the same cancer, but it has become chemo-resistant. Now you want to understand why these cells have become chemo-resistant. A good way to do it is to study their proteome. You study both these proteomes. Cell line A, cell line cell line B. Same treatment, one is chemo-resistant, somebody is working on drug resistance, evolution of drug resistance in microbes. So suddenly you find the particular antibiotic resistance is developing, some antimicrobial resistance is developing. So you take both the proteomes and then you want to compare. So this is for exploratory investigation. So you know how explorers worked, how the British worked. They set sail with their ships, and whichever island they hit, they told this is ours, and they suddenly discovered things. So that is how discovery works. You discover. So until and unless they discovered India, India was not there, or so they believe. So that's the joke between me and Professor Graham. So so fact of the matter is, that is how discovery works. Something was already present, you did not know it was there, you used a technique and you found out. Okay, this is what is happening. Then what is targeted? Here the question is, are proteins A, B, C in my sample? That means you want to probe that whether your friend went out with another friend last night, right? So you would try to ask other friends, देखे थे इन दोनों को? वही थे ना? अच्छा, कॉफी पिए हैं? आइसक्रीम नहीं था, कोल्ड कॉफी थी. So now you are probing, now you are asking very targeted questions, right? So that's what you do. Suppose you want to, in a particular sample, you want to find out whether protein A is expressed. Now you don't want to know the differentially expressed proteome. You just want to know how much of protein A is being expressed, how much of protein B is being expressed. So it's more like Western blot. But we will come to it, why we would use mass spec and why not Western blots. Because Western blot, those who have a proper idea, I think many of you would have known what is a Western blot or an immunoblot. It is a very good way of telling that it is targeted proteomics, but only fact of the matter is, since it is not a high throughput method, is if you can do 100 Western blots in one day, that would be targeted proteomics. Whereas discovery proteomics is primarily studying the entire proteome, and ultimately it leads to tell things like differentially expressed proteome or binary proteins. And what are binary proteins? Binary proteins are proteins which are present in one group, absent in the other. Right? Binary, non-binary. Like nowadays, in the US, you have binaries and you have non-binaries. Binaries are like male, female. Non-binaries are gender fluid. They could be anything. Morning, female, afternoon, male, evening, female again. So, non-binary. No, I'm not kidding. You can just... But yes, you can have a laugh. So, these are primarily for hypothesis-driven investigation. So Professor Brown was telling yesterday that sometimes we do not need a priori hypothesis. When you are doing a discovery proteomics or exploratory investigation, suppose you want to study the impact of a drug on brain or impact of drug on a specific cancer or any system, and you want to explore that what all this drug could do, then it becomes exploratory proteomics. Now you want to understand impact of this drug on this particular pathway, where you will probe only for those specific pathway. Means your hypothesis is already set that this drug might alter pathway ABC. So my drug X works on pathway A, B or C, and this pathway ABC would have 1, 2, 3, 4, 5, 6 proteins. You use a mass spec to probe those 6 proteins. What you can do with this is, you can do relative and in fact absolute quantification. You can do absolute quantification or quantitation. You can absolutely measure the amount of protein each of these groups have. This shotgun proteomics, primarily nowadays the LFQ based proteomics, can give us relative quantitation, where we tell this is like fold change, 2-fold change, 3-fold change, but with respect to what? We do not know, right? I'm 2 times better than you or 3 times worse than you. That's the ratio. That does not tell us how much of the protein is present, right? When you do absolute quantification, you can tell 100 nanogram of protein X was present, 1,000 nanogram of this protein Y was present. And this is very important, especially when you're trying to develop a diagnostic assay, right? You have to do a LOQ. What is LOQ? Lower limit of quantification. So you have to make, when these kind of assays, the targeted assays, they have limits of quantification and stuff like that. So, and one more idea is why we need to, I think again I'm repeating what Professor Sanjeeva said, so there will be lots of repetitions as well. So if you look at genes, almost 22,000 genes, again new news is it's 22,500, things are dynamic, but when you look at proteoforms, which are isoforms of proteins, right? Same protein, if it is phosphorylated at position 396, becomes a different protein, right? Suppose one protein has 7 phosphorylation sites. That one protein can, how many, now tell me what could be the possible, like a simple example. Suppose there is one protein that can be phosphorylated at 2 sites. So that protein can take up 3 proteoforms. 1, it's completely phosphorylated. 2, it is half phosphorylated. 3, fully phosphorylated, and within the half phosphorylations it can take up 2. So actually even with 2 isoforms, 2 phosphorylation sites, you can get 4 different types of proteoforms. That, and due to the structure function relationship, these 4 proteoforms can act in widely different ways, right? Cell division cycles, kinases. Look, if you go back to cell division cycle, you would see how phosphorylations can drive the cell cycle from G0 to G1, whether it will be question. No mumbo jumbo here. What is normative aging? Forget aging and cancer, aging and gender. It's just how do we age? Right. So there we will do systematic proteomics. We will do proteome profiling. We create a proteome profile that tells us that, okay, this particular cell has 6,000 different proteins. End of story. Now, then we can do differential proteomics. Here we can compare proteome profiles in different physiopathological conditions. Here you can bring in most of the proteomic study that you would find up if you look up on PubMed is differential proteomics. Impact of nerve gas, impact of cancer, impact of turmeric on disease XYZ, on brain, on liver, impact of COVID. That's what we did while we were studying COVID. So what happens in severe and non-severe COVID? So differential proteomics, we will compare the proteome profiles, maybe that of an organ, maybe that of anything. Like somebody was in the morning was talking of bacterial cells, chlorella. So it can be from chlorella as well. We just want to understand the differences. Then there is, and what does it tell? It can identify disease-specific biomarkers. If the proteins are extremely different, you see that protein X was not present in one condition, or it was faintly present, and suddenly it has gone up 25 times or 30 times, then immediately it becomes a biomarker. But only when you have a fair number of sample. It's not me, right? It has to be this, in a population, at least 95% people have to have that same protein going up, only then it becomes a biomarker. Next, the structural proteomics, which of course is different from bottoms up proteomics, but since you are into a proteomics lecture, you should understand that we use proteomics to study the structure. So people who design biosimilars, how many of you are aware of the term biosimilars? Yeah, what is biosimilar?
It is like a generic version of already marketed biological formulation.
Right. So you must have heard of tocilizumab and all these kind of antibodies that are used for therapies. Now, when you manufacture such a antibody, now antibody is a big molecule, right? And you need to characterize it. You need a proper structural study of it. So there are ways in, not of course not by bottoms up, but by a top-down approach. You can do structural proteomics as well. Then you can do functional proteomics, where you want to understand both by systematic profiling or via differential profiling how the subcellular proteins change. So you want to study the cell function. Now you would like to study the proteins at the nucleus, the proteins in the mitochondria, proteins in the cytosol, proteins on the cell membrane, right? Cell surface proteomics. That's also one way of doing it. Then you have metaproteomics. People who are trying to study, many of you would have heard the word metagenomics. So almost an equivalent term is metaproteomics. So metagenomics, there is an amplifying step. In metagenomics, there is no amplifying. That is good and bad at the same time. The reason is, in metagenomics, since there is lots of amplification done, sometimes you amplify noise. Here, since in metaproteomics you don't do amplification, the noise is cut off, but this is bad as well, because sometimes even if the signal is faint, you don't get any signal. I mean, if the number of proteins present for a particular microbe is less, you don't get any good amount of signal. So all these together can help you to understand protein-protein interaction, the mechanisms or functions of proteins, how proteins are moving around, compartmentalization of proteins, how proteins migrate. All these things can be studied. Also, an impact of microbes on gut ecosystem, the gut-brain axis. That is you can all do that from the proteomic data. Sometimes you can also take up genomic data and do something known as proteogenomics. A good example is, suppose you have anybody is suffering from cancer, right? What is a cancer? An aberration in the genome, and that aberration in the genome most likely has altered the proteome. That made the cell go crazy and divide like there is no end of day. It keeps on dividing. If we could understand for that particular cell what is the genome, then we would also be able to understand what is the proteome, right? That entire thing becomes proteogenomics. Very important when it comes to cancer. Apart from that, there are also terms like immunopeptidomics, where you look at the immune cells, the MHCs in particular. If you remember how MHCs function, you would remember that there are small parts or small peptides that the MHC molecule holds on to, right? That are actually coming from the antigen presenting cells. Now, using immunopeptidomics, you can also understand what are those peptides that are being antigenic. So you can also understand how your body immune system is responding to sometimes even to cancers. That's why people do a lot of immunopeptidomics in cancer. That again can be translated to something known as CAR T cell therapy. Those who know it, else you can just look up on it. I think we will take a small pause. You can have tea for 5 minutes and come back. The tea came 30 minutes late, so sorry for that. Webcasting team, you can pause for 5 minutes if possible. Please have tea, and we'll be back in 5 minutes. Since we will be taking a break, we have to be there. What we can do is, first half we will have an exam, maybe of 20-30 minutes. That's on Google Sheets, right? Open the QR code. I will ask my volunteers to turn it on maybe at 10:30, then turn it off at 11. Be honest with yourself. You can obviously cheat because it will be on Google Sheet, so you can open ChatGPT and start sharing the same questions. But again, anybody who gets full marks also does not get the certificate. Right. Beyond that, there are a few tools that you need to understand. One is MaxQuant. Somehow things are not working as per schedule. So MaxQuant is a tool by which you can identify the raw MZ values that you get from the instrument that can be translated into proteome. So I will share a slide with, maybe if in the early morning, how much time do I have? I think 11 baje aana hum logo. Right. So before 11, I will give you brief description of MaxQuant, MetaboAnalyst. These are two tools. Then I will share few already published data sets that we have with us. What you can do is, MaxQuant will take time, and it might not run on your PC. It will take 2 hours. So you can just go through I will tell you how MaxQuant works, what's in the hood. Remaining, we will give you a raw output of MaxQuant that I think Dr. Shambhu, about to be Dr. Shambhu, can explain to you. And beyond that, we can study Metascape and Reactome. So that would be for tomorrow. We do not know how long it will take for us. Although it's a 1.5 hours program, few issues need to be sorted. Since the Chief Minister is coming, we have to be there early. Since the Chief Minister is coming, there will be a lot of vehicles. The moment we come out of that campus, despite being 700 meters away, will take few, God forbid. So after 11, most probably on a very good estimate, we will reach by after lunch or by lunchtime. So we will keep you engaged in that way, and that will be third day. If you have any specific queries or you want that few things need to be repeated, Professor Graham's lecture handouts I will be sharing. My lecture handouts I will be sharing. What all do you need? Professor Sanjeeva's I have asked for him, but if he shares I will share. And have tea, then we can discuss the rest. Right? Most of the but gel based has its own benefits, like we were discussing in the early morning for the first session in the morning, that how gels are a very cheap way to achieve things that even a mass spec cannot tell you. A mass spec can never tell you whether your proteins were intact or degraded. If your protein, costliest mass spec cannot tell you. It will only give you some data which you won't be able to make sense out of, and then you would realize it was not good. So this part we have already taken care of, right? Samples, protein extractions, protein separation, and this separation based, so basically gel based and gel free proteomics primarily is a separation technique. You have to separate. You have lots of proteins, you have to separate the proteins. How do you do it? So how many of you heard about DIGE? Anybody know what is DIGE? You know 2D, right? So 2D is what? Two-dimensional electrophoresis. Dimension is isoelectric focusing. The second dimension is based on the mass, right? So first one is based on P isoelectric. The second one is based on the molecular mass. Now the problem with 2D was when you have to compare across specimens. Suppose you have 6 specimens of breast cancer and 6 peritumoral tissue that basically you take from just beside a tumor. Now if you have 6 different tissues, how many gels do you have to run? हाँ बोलो. Department of Geography, Anupam Pandey. Anupam Pandey, Department of Geography.
Okay sir, thank you.
हेलो
हाँ बताओ.
सर दे चुके हैं, अभी आ रहे हैं, हम लोग बहुत थक गए, अंदर गाड़ी भी नहीं आने दी, पैदल दौड़ रहे हैं.
ओहो. चलो चलो थोड़ा weight loss. And that I got in GAN was, if you are an admin, don't talk. Don't have your own talks scheduled. Get somebody else to do the talking. Anyway, so as a teacher you can't stop yourself from teaching. That's again. So if you have to have just 2 types of tissues, cancer, non-cancer, to achieve some degree of robustness in your data, how many gels do you have to run? One gel and you get something, you would find a biomarker with just 1 gel? 2? No? So there is something known as biological replicate and technical replicate. Biological replicate is usually for these high definition data sets is usually set between 8 to 10. Means you have at least you have to take 8 to 10 individual samples. So 8 gallbladder cancer samples, 8 normal gallbladder, right? Technical replicates these kind of, so that is biological replicate. Technical replicate is triplicate, 3. So 8 multiplied by 3, 24. So if just for control you have to run 24 gels. Similarly for disease you have to run 24 gels, 48 gels. Now tell me what is the what's the chance that all these 48 gels will be run under identical conditions, nothing going wrong from your side, from the electricity supply side, everything remains perfect. So there are lots of areas where things can go wrong, and things did go wrong. That's why this label-free approach was dropped. We took up a labeled approach that we call as differential in-gel electrophoresis. So DIGE, we label 3 samples. You can see the red, green, there are 2, okay? So you have labels, Psi3, Psi5, these kinds of fluorescent labels are labeled. So when you label these samples, suppose the control sample was labeled with a blue dye, with blue fluorescence. The treated was labeled with red. And then you usually run another sample that is a mixture of both the two, and that we label as green. Now can you run the same gel on a same gel, because all these samples are now labeled with fluorescent tags? Just by scanning one gel at 3 different wavelengths, you can get results, right? So that way you are reducing the number of gels you have to run just by half. From 48, you drop it down to 24. So this is DIGE. You can look it up on DIGE, and after DIGE what you do, you pick spots. These spots are those spots which shows differential expression in protein. Of course we go for trypsin digestion, which I asked you not to use, but again this is a layman's term that most people use, tryptic digestion. And then you do ESI Q12 NS, which is ESI stands for very good, electrospray ionization. And what is TOF? Time of flight. MS means mass spectrometry. So, and then you detect ions, then via database searching, which basically MaxQuant will does, a software like MaxQuant will do for you. You can identify the proteins. Same thing that you can do in LC based proteomics, in liquid chromatography based proteomics. And in liquid chromatography based proteomics, it can be either labeled or label free. I think I was over here, Dr. Bagel, he was discussing about different kinds of labeling approaches. What kind of approaches did he discuss? So there are multiple types of labels. Maybe if you have time we will discuss. So there is TMT labeling. TMT stands for tandem mass tags. Then there is stable isotope labeling that we call as SILAC. So if you use it for animal culture, it's called SILAC, animal cell culture. If you are using for live mammals, you call it SILAM, stable isotope labeling for alive mammals. And what you do is you take isotope labeled amino acids and put it in the media, so that when your cell starts growing, those isotopes are taken up, and then when you do the mass spec, you can identify just based on isotopes that what was my cell. Suppose you have, so these are label based technologies. Label based technologies have issue, especially there is also ITRAQ, ITRAQ. So ITRAQ based labeling, TMT labeling, SILAC labeling, all these are labeling techniques. There is always an issue with labeling techniques, but labeling techniques are not completely reliable. They have around 70% efficiency, and all proteins do not take up labels. Suppose you have used a specific amino acid, you won't label all the amino acids, right? You take 2 or 3 amino acids which you label in SILAC or SILAM. Depending on the presence of that amino acid in your protein sequence, your labeling efficiency will vary. Suppose you have taken valine or alanine or tryptophan. and you have started labeling every tryptophan. You have taken the heavy tryptophan, and your tryptophan wants to get incorporated into the cells. It will only be incorporated at specific where there is a codon for tryptophan. Suppose your protein has very few codons for tryptophan. Will it be labeled well? No. Second issue is, suppose it got labeled well, but when you go for the trypsin digestion, the peptides that are generated are either too big or too small. So you might end up with a big zero. That's why people slowly moved on to labeled methods, where label-free methods, where just by analyzing the chromatogram, you can, so much of the work that is being done these days is label-free. In label-free, all you need to do is, we will discuss in detail today, we are running out of time. So in label-free, what you do is, we don't label the proteins as such. We take the proteins, we make peptides out of it, we inject the peptides, and then based on the chromatogram, we try to identify what this protein could be. So LC-MS tandem mass spectrometry coupled with an LC helps us doing this. So also here also we use a ESI QTOF-MS. Sometimes we use Orbitraps. Then there is another method called CELD. Now CELD is actually something of a very old technique, but we don't use it anymore. So again, getting back to what is the basic composition of a mass spec. We have a sample, you have an ion source, you have a mass analyzer, you have a detector, and you have a data analysis. So mass spectrometry is basically this. Now it could be a hybrid instrument, it could be a tribrid instrument, it could be any instrument. All depends whether you have one or multiple mass analyzers. So a hybrid instrument will have two. A tribrid will have three. An ion trap and Orbitrap together, that makes two. Sometimes you also have a TOF thrown into it. The latest Astral is basically hybrid plus TOF. That gives you more sensitivity. So what does the ion source do? Mostly by electrospray ionization, which is a soft ionization technique, or by MOLDI, that is a matrix-assisted laser desorption ionization. It is a very hard technique, but in both techniques, you generate ions. You make charged peptides. You charge the peptides, and once the peptides are charged via the ion source, it will go to the mass analyzer. They will do a detection. They will analyze the mass. The detector will read the signals. Now I don't want to create a lot of fear in you, but there is a lot of physics and maths going around here. So usually we do, in an Orbitrap, they do a Fourier transformation, right? It's called FTMS. That's the reason sometimes Orbitraps would have FTMS, Fourier transform MS. We won't get into that. We will keep the biology side heavy. So we detect the different mass by charge ratios, and then the data analysis part, which we will take up tomorrow, which we're supposed to take up today, we will take up tomorrow and decides. So again, if you basically divide mass spectrometry in today's time, there are discovery-based and hypothesis-driven workflows. Discovery-based workflows do not have any a priori hypotheses. You just want to understand how these things work, right? It is unbiased, and we do proteome or post-translational modification, quantification. We just quantify qualitatively and quantitatively what is the proteome. कितने proteins हैं, how much of it. Sometimes we do relative quantification. Usually it's good for and what we do is primarily the BDA and DIA. I guess Professor Sanjeeva also hinted upon it. So there are two ways by which an instrument can pick up signals. to give you a very rough idea of how these things work. Suppose some ions are, you generate a lot of ions, right? In data dependent acquisition, the larger the ion quantity, the more the number of ions, the more easy it will be detected. So that is data dependent. Data independent and in DIA based methods, this is not a challenge. So in DIA methods also you need DDA to create a library where the I'm not going into the details, but thing is DIA is dependent on DDA. First you have to run a DDA to generate a library, ion spectra, and then that ion spectra will be used for DIA based methods. DIA methods, since we are not basing just by the number, the sheer volume, so these processes are very democratic, right? DDA is extremely democratic. More the numbers, more easily will be found. DIA is more like the Rajya Sabha, where you already have few selected members, you have a library, so that's how Rajya Sabha people are chosen, right? So we want to have a balance. So in Rajya Sabha, people come from the legislative assemblies, people are nominated by the president. So DIA is more like that. It gives you a better coverage because you can shut down the noise that is coming from some abundant proteins. Hypothesis driven or targeted studies primarily work via two methods. One is the selected reaction monitoring or parallel reaction monitoring, SRM and PRM. And what we are seeing here is primarily three different types of instrument. One is primarily designed for DDA or discovery based studies, where you see this is a triple quadrupole. This is a mass analyzer, right? This is also a orbit trap mass analyzer. So these are all mass analyzers. This is either by DDA or DIA. Then you have a targeted type of studies. We can also use orbit trap for doing PRM, parallel reaction monitoring. For SRM, we use these kinds of triple quads. So there are these, one will select specific ions, then we will break the ions, we will come to it. So you can just remember for the sake of now that there are multiple methods. One is hypothesis driven, which is targeted, another is unbiased, discovery driven. And how does this entire process work? This part we have already discussed. We have come till peptide, pure peptide. Then we have electrospray ionization that creates molecular ions. Then we do the first MS that gives us the single M by Z. Then we do dissociation, where we break down the ions further, that gives us A ion, B ion and Y ions. This is very essential because we want to sequence, we do some shotgun sequencing of the protein based on the splitting of the protein peptides. So we do the second MS, the MS2. This creates the mass spectra. This mass spectra comes into the search engine like Andromeda, like tomorrow we will discuss what is Andromeda and stuff like that. To look at it, there are three levels of it. First is wet lab operation. Any lab who is perhaps passionate about it can do the wet lab. Now, BHU has Sathi Center, Sophisticated Analytical and Technical Help Institute, Sathi. So BHU Sathi can help you with that. Also at IIT Bombay there is a MASFET B facility. At CDRI maybe most of these facilities are central facilities because these are very pricey instruments running into crores, not lakhs. It's 2 crores, 3 crores, 12 crores. Then once they generate the MS spectra, this part again can be done in your own lab if you have a decent computer, a desktop with maybe 16 GB of RAM or 64 GB of RAM. Yeah, that's decent. 4 GB is not decent. So then you can run multiple spectras, and nowadays there are much smarter softwares, MS Fragger and stuff, which can run on much lower computing needs, and that can give you, so the middle part you can outsource, top part you can do in your own lab, does not cost much money. I mean, it will cost in lakhs of course, but not in crores. And again, the computer operation, it's a one-time investment. If you can get the tools that Professor Graham is showing, with that you can sit at your home and do your own research. It's mostly DIY. So what can contemporary proteomics do is, even with the very cells and tissue, it can identify 2,000 to 3,000 proteins. I'm not even going to timsTOF, which can go to 10,000. Even an average Q-Exactive will identify almost 3K proteins. It will give you a very precise relative quantification, that is targeted proteomics. You can also identify different types of post-translational modifications. Very important if you want to study signaling pathways, right? Phosphoproteomics is a big thing. Many people working on cancer, just by looking at the proteome, won't get much of an answer. But the moment you look at the kinome, they have very fancy names to it. Like people call it the kinome because it is driven by kinases. So if you look at the kinome, it can tell you what kind of proteins are getting activated, because multiple enzymes, if it is an enzymatic protein, it might depend on whether it is being phosphorylated or non-phosphorylated. Also, if you want to identify how proteins are interacting, you can do co-IP, co-immunoprecipitation, and then with those co-IPs, again can identify how many protein interactors do you have, right? So in co-immunoprecipitation, this is a very old use of mass spec. I have been hearing it about, because you don't need to be very high throughput for this, but even with this, a good co-IP, you can give up actual physical interactors to your protein. So I guess we are almost on time, just that we had one less lecture. But anyway, Dr. Anil's was an addition. So I have one more lecture pending, so tomorrow maybe morning we can start with that. 9, we can stop the webcast.